afternoon, everyone. I'm going to talk about natural language processing in text mining for protein docking. So protein-protein interactions are very important in various biological processes. Due to limitations in experimental techniques, protein docking is the key to understand protein-protein interactions which are crucial for cellular mechanisms. There are some, in high throughput techniques, we get high proportion of false positives. And therefore, protein docking is important. But an obstacle to success of protein docking is unreliable discrimination between false positives and near natives. Therefore, constraints are important. It is possible to get information about protein-protein interaction sites using text mining. We have developed a text mining tool on bound protein-protein complexes and validated it on unbound protein complexes taken from dog ground. Basic text mining has shown us very promising results, but it suffers from high rate of false positives. And to make text mining more meaningful source of knowledge in protein docking, it is important to enhance this further. And therefore, we have added another layer, which is an extension of text mining tool, the natural language processing NLP technique, to reduce the false positive with respect to residues. In this plot, you see in the blue, the bar is low resolution geometric scans taken from ab initio gram docking without post-processing. In the red, when the text mining constraints were added to it. And in the green, this is the reference, and this is the best we can achieve because these are the actual residues in the bound complexes. And this is the maximum we can achieve because this is rigid body docking. In the bottom plot, we see on the x-axis is the correctly predicted by predicted residues. Correctly predicted by the predicted residues. We want the bars to be towards 1. 1 here means all the predicted residues are correct. 0.5 means 50% are correct, and 0 means all are incorrect. The system architecture of our text mining tool is given two proteins. We get the protein names from our CSV website, from UniProt, and once we have the proteins, we form two kinds of queries, AND queries and OR queries. AND queries means both the proteins are mentioned in the abstract, or means only one protein is mentioned in the abstract. Once we have the abstract, we look for residue name and number, like serine 4, SER 4, such kind of patterns. And once we have that, we give higher confidence value to AND query residues over OR query residues. And then we use these as constraints in docking. And now we have added an NLP layer over it. We explored various NLP procedures at a sentence level, going from WordNet to dictionary lookup, where we use fact of interaction words, and then going from syntactic to semantics of a sentence using deep parsing. In WordNet, what we have done is we took the sentence which has the predicted residues, take the each token or the word, and then use various similarities measure to get the closeness of each token or the word to the closest synset of contact, which is respect to binding or site. Then further, we went from a general English purpose WordNet to domain specific here, fact of interaction. In the bar we see on the x-axis at 1, we get the bars which are taller, but then the corresponding bars are also tall at the 0. So we realized the problem here was our domain is even more specific. We had to go from fact of interaction to mode of interaction. And therefore, we went to deep parsing. And this is the last part, in which we take the sentences, the embedding sentences which has the predicted residues, get the parse tree, and the key assumption here is words closer to residue in a sentence are more relevant than further apart with respect to the parse tree, and we take the distance between the residue and that of the entities, that is the mode of binding entities in a parse tree, and we also take the context of the surroundings, that is the surrounding sentence, For the surrounding sentence, we take the parse tree with respect to the root. We also take other features like presence of proteins. We add all these features in the feature vector, SVM, and then we try to differentiate between protein-protein interaction sites and non-protein protein interaction site. And in this result, we see that deep parsing performs better than the AND query with respect to accuracy and recall. With this, I would like to acknowledge my advisors, Professor Ilya Vaxer, Professor Petros Kondritos, and the University of Kansas. And my poster is today, it is L11. Thank you.